everyone. Thanks so much for joining the virtual IBSS seminar. And thanks to Natalie for setting everything up, all the logistics, and Catherine also. My name is Rebecca Johnson, and I co-direct the Citizen Science Department at the Cal Academy, for those of you who are joining from outside. The rest of our team includes the other co-director, Allison Young, and then Annie Miller, and then Dr. Gio Rappucciolo, who we're going to meet right now, who will be talking about his work monitoring California's coastal biodiversity with citizen science. And Gio comes to the Academy and really adds, when he first arrived about a year and a half ago almost, just added a whole bunch of new skills and ways of looking at things to our team, which have been amazing for this project, but also I think for our whole department's work. And it's been just a joy to have him as part of our team. His work really focuses on modeling past and future impacts of environmental change on the distribution of species and ecosystems. And he really made a big shift when he came to the Academy, because he really cares about bringing the macroecological tools that he uses to conservation practice. And I think you'll hear a lot about that in his talk today. Gio got his PhD and his master's in biodiversity and global change from the Imperial College in London. And then he did different postdocs at UC Merced, with NatureServe, and at UC Berkeley. And I am super excited to hear about his work that he's presenting today, which is kind of a culmination of what he's been working on for the past year and a half. So without further ado, I'll turn it over to Gio.
Thank you. Thanks, Rebecca. Thanks, everyone, for tuning in. I can't see you, but I can imagine you. So yeah, thanks for tuning in. So I'm going to dive right in. Thanks for that great introduction. And as you know, we've entered a new era, a new geological area, the Anthropocene. And the Anthropocene is characterized by the pervasive influence of humans on Earth processes. And one of the unfortunate things that are happening out there, that biodiversity is under great threat. And the big 5 drivers of biodiversity threat and biodiversity change are these ones here in the figure. So land use change, harvesting, climate change, pollution, and invasive alien species. And these 5 big drivers are characterized by being global. They don't care about geographical features or borders. And they are dynamic, they change rapidly, and they evolve through time, and they are pervasive across the tree of life. So they affect many species, and they have cascading effects across the tree of life. So if we are to hope to try and understand what the impacts of these 5 big drivers on biodiversity might be, we need big data. And we need data that are big in all 3 of these dimensions. That is space, time, and taxonomy. And so you can think of this as the space of ecology, or when you're large in one or all 3 of these dimensions, the space of macroecology. In its basics, macroecology is large-scale ecology. And so the reason we need to be big in one at least, or all 3 of these dimensions, is that then we get to explore the full range of variation that biological systems have been subjected to throughout the globe and across the entire history of the world. And so we can better understand how the environment influences biodiversity, and has in the past and is likely to do in the future. So as an example, this is a recent publication where we tried to figure out just how far in geography and how far back in time you have to go to understand a community in a given locality at the present. And our conclusion here is that you potentially might have to go back millions of years and look on the other side of the globe to try and capture processes like evolutionary change and long distance dispersal. So where do we find the big biodiversity data to explore variation over space, time, and taxonomy. If you go over to GBIF, the Global Biodiversity Information Facility, which is really the warehouse for big biodiversity data, but it's also so much more than that. It's a global network of partners, but it's the reference for global biodiversity data. If you go out there, you'll find that over 50% of the data that are in there are citizen science observations. And 6 of the top 10 data sets that provide data to GBIF are citizen science data sets, including the Academy's very own iNaturalist. So you can see it down there. And so the main point here is that we really ought to figure out how to use citizen science data, how to work with these data to be able to understand large-scale environmental change and its impacts on biodiversity. So this is where our project comes in. The Ocean Protection Council, which is the California agency tasked with the protection of coastal and marine areas in California, had the incredible foresight really to understand that these data may be useful for monitoring purposes and to generally better understand large areas like the California coast. And so this collaboration with the Ocean Protection Council and the California Department of Fish and Wildlife is what I've been focusing on over the about year and a half almost. It's drawing to a close next month, and it's going to be the subject of the rest of this talk. So what are the citizen science initiatives at the California Academy of Sciences that we're talking about here? Specifically, you'll be familiar with them. The first one and the main one that really gets people out to the coast and is really the main engine of data collection is Snapshot CalCoast, which was started by Rebecca Johnson and Alison Young, my co-directors at Citizen Science. And Snapshot CalCoast is an annual statewide effort to document coastal biodiversity. Essentially, it's a series of coordinated bioblitzes up and down the coast to try and document biodiversity as well as possible over such large scales. And the other one, which obviously is intimately related, because during Snapshot CalCoast, data are collected using iNaturalist. So iNaturalist, I hope you know it. I hope you have it on your phone. Is this amazing tool also developed at the Academy, co-directed by Scott Laurie and Kenichi Ueda, is an amazing tool to record biodiversity by essentially taking a picture of it. And by taking a picture of an organism wherever you are, you essentially take a picture of something and give it a timestamp and some coordinates. And that provides the basic data that you need to understand the distribution of biodiversity and how many different organisms are where. So I don't want to spend too much time introducing these things, which we'll be fairly familiar with. But this GIF right here shows the incredible growth of iNaturalist records on the California coast over the last 10 years. And you can see that there are now upwards of 50,000 or so records a year. And you'll see that that peak is actually a combination of Snapshot CalCoast, which I was talking about, as well as the City Nature Challenge, which kicks off tomorrow. And both those initiatives generate a huge burst of observation on iNaturalist, and that includes on the California coast. So that's where most of our data come from. And these are research-grade observations. We'll talk about these a little bit more in depth in a minute. So the objectives of this project then were to, and are still, to provide recommendations for increasing the usefulness of ongoing citizen science data collection up and down the coast. specifically for MPA management. So that's marine protected area management, but generally to understand coastal biodiversity. And so the second objective going on from that is to generate knowledge of California coastal ecology and better understand the effects of environmental changes on coastal biodiversity. So the big question then is, how do we extract signals of biodiversity change from citizen science data? And I've put this word here, unstructured. These data are unstructured in that they are not collected using any specific type of survey structure. There are no instructions that you're given for providing iNaturalist observations. The minimum common denominator is you've taken a picture and your phone has assigned it some GPS coordinates and a timestamp. So let's think about these data a little bit. There's recently a unifying framework that has emerged for thinking about these data, and they're not exclusively just citizen science data. These are just data that have those basic features I've outlined: a timestamp, some coordinates, and an identification of what organism it was. So point process models try to understand these data by thinking about them as a point process. So we have this figure right here shows you can think of this as a series of individuals across a landscape, and this here, lambda, is the intensity, or you can think of it as the density of individuals across this landscape. It's higher in the green here, and then it sort of decreases as you move up. So generally, if you were trying to understand the variation in density of individuals of this species across a landscape, ideally, you would do something that's called a standardized survey, and that is randomly you would throw some quadrats, or you would look at an area, specified areas across this landscape, and then count or attempt to count everything that ended up in that square, all individuals that ended up in that square. If you do that, you can basically analyze those data fairly simply by taking those at face value and just looking at how the intensity here, or the density, varies as a function of some predictors, let's say temperature, for example. We're interested in the effect of temperature on density. You could do that at face value. The challenge is that with data like citizen science observations or point occurrence data, species occurrence data, we have a biased presence only type of sampling. That is, we don't have squares laid out randomly across the landscape. We actually have a series of observations that correspond to some of the individuals across this landscape. So these are the blue triangles here. And ideally, if those blue triangles, those individuals, had been observed randomly across the landscape, then we would have something that approximates a survey design. Unfortunately, the observations are not taken randomly. And so as you see here, we don't have an estimate of the density. If we look at these data at face value, we have an estimate of the density times this function here, which is the bias. So we have a biased estimate of the species density, and we cannot analyze it by taking the data at face value. We need to add the second part here, which is concerned with understanding bias and includes predictors of bias. So how exactly are citizen science data biased? And through the slide here, I just wanted to show you that the biases that we're talking about are actually very similar to other types of data and other data that we're very interested in, like museum specimen data or herbaria data in this case. This is a publication that shows that herbarium data biased by things like human population density, the distance to herbaria. So the closer to the herbaria, the more the observations, and things like distance to roads, so accessibility. If we have a look at iNaturalist data and citizen science data, we find that many of these things also apply. So on the left here, you'll see the pattern of density of iNaturalist observations. This is a logged pattern, but so this here on the y-axis is that variation on that map. And here on the right, you have a map of a heat map of the density of coastal access locations. So those are places that you can go and then access the shore. And specifically, in fact, those are coastal access locations that have a parking. So you can actually park there and access the coast. And as you see, there's a pretty strong linear correlation between how many iNaturalist observations we have along the coast and whether you could access the coast there with a parking lot. So the first issue is that iNaturalist data are uneven. They have an uneven coverage in space, and they are biased by things like coastal access. They're also spatially biased by things like population density, again. So this is a map on the right here of the population of densely populated areas. And this is the correlation between the number of observers and the density of population centers. Unsurprisingly, there are more iNaturalist users in high-density populated areas. The other bias that we have in iNaturalist is an uneven coverage in time. So that means that, fortunately, more and more people are using iNaturalist and signing up, and that's fantastic. So this is this curve here on the right, the cumulative number of iNaturalist users over time, which is going up fast, actually, almost exponentially here. It's not a linear increase. But what that means is an almost equal increase in the main phyla here on the coast. This all applies to the coast, by the way, the area that I'm showing you. So this is how the observations of the main phyla on the California coast is increasing parallel to the number of users. You can see, however, that some phyla are maybe tailing off a little bit, which means that we may be actually getting to record more and more and getting an exhaustive sampling of some phyla. That doesn't apply to mollusks, which is a very high diversity taxon. The third and main bias that I will be talking about, and really the third and main bias all in all, is variation in observation effort. What that means is that not all visits, not all iNaturalist observation events are as comprehensive as each other. And so on the right here, you can see that on the y-axis, there's the percentage of visits over years. And these are colored by how many species were observed in that visit. We'll talk about what a visit means a little bit more in a minute. But for now, just think of it as a single iNaturalist observation event by a single person in a single window of time and at a single location. And what this is showing here is that the vast majority of those events, almost 3/4, between 2/3 and 3/4, generate one species. They involve the observation of a single species. And less of those involve observing 2 species, three species, and then 4 species or more. We do, however, have like a good chunk of data here, and it's getting better as well, which is probably comforting here. But we probably have 20 to 25% of events that generate 4 species or more. And this is the density here on the left of high intensity observations across the coast. So you can see that the Bay Area, and in fact, this here is probably Pillar Point right here, which has a very high number of visits that generate 10 or more observations. This is the number of observations, not necessarily how many species those observations generate. So the issue basically is that we cannot use these data at face value if we want to extract signals of biodiversity change. because what we really would be extracting is more the behavior of observers on iNaturalist and what they choose to observe and how frequently they choose to observe it, and the fact that there are more and more of them observing these days. So those of you who know me know I love cooking, so I turned the steps that I found over the last year and a half can help increasing the usefulness of these data and teasing out actual ecological signals from them. I turned those into a recipe here that includes 5 steps. Unlike a recipe, though, however, don't think of this as a set of sequential steps, but rather there are steps that may all apply or not, and you could use them one after the other or in combination, but they are a set of useful steps to account for bias in unstructured citizen science data. So I'm gonna go and discuss these and give you examples of how I've teased out some signals out of iNaturalist data using these steps. So the first one is perhaps the most obvious one, maybe, and it involves filtering the data. You probably cannot use all of the data that are in there. And for instance, the first step is to try and reduce measurement error. iNaturalist data and citizen science data, like any type of data, are subject to measurement error. So, for example, the spatial coordinates may not be so precise, or the timestamp may not be so precise, and maybe the taxonomic identification isn't right or is subject to uncertainty as well. So amazingly, the iNaturalist team takes care of a lot of that by assigning observations to the research grade category, which tries to reduce the uncertainty in measurement error. And so from now on, I'll be talking about research grade observations only, and I direct you to their amazing blog on iNaturalist for more discussion on how research grade observations actually reduce measurement error. The other part here of filtering data is to try and start to even out observer effort. So the biases that we were talking about earlier. And so what that means, for instance, I'll give you an example, is that instead of looking at things that have huge differences in, say, the number of observations and number of observers, we can look at subsamples of these areas or times that actually include the same number of observations or same number of observers. We can reduce the side, if you want, that has higher observer effort to even out observation effort. I'll give you an example of it. This is an example of change in the community composition, the ecological community composition over time across 5 different counties. So the filters applied here are, once again, they're all research grade observations, and those are all counties that had at least 100 observations every year here. So there's a filter for a minimum amount of information here. But on top of that, we're not using 100 observations and however many there were. Oftentimes there are many more than 100. But what we're doing is, in case there are many more than 100 observations in any given year in any given county, we are actually choosing a random subset of those to equalize the amount of observations that we're comparing year on year. Because obviously by choosing a subset, you're losing out on the other observations that are there. You don't just do it once, but you do it over and over. So you choose 100 observations randomly, and then you do it again, and then you do it again, hundreds of times, and then you look at an average pattern between the set that only had 100 observations, say, and the higher set that had many more subsampled in that way. I hope that made sense. Please ask me questions about it if that didn't make sense. I'm just trying to give an overview here of a number of things. But when you do that, you compare the ecological composition. So that's what kinds of species and how frequently they were observed over time. you can start seeing differences in the change year on year in the composition of species. And as you can see here, there's something maybe going on in LA County, which saw a very big change between 2014, 2015 here and 2016, which means that here there was a big change in the kinds of species that were observed and how frequently they were observed. Once again, this has taken care of some of the sampling biases I was talking about earlier by doing the subsampling. Same thing again here. I just wanted to show you the pattern between regional, coastal regions, sorry. So that's the North Coast, the Central Coast, and the South Coast. And what's interesting here is that even though those differences may not be huge, we are seeing generally, especially around 2014, 2016, which was a year of strong temperature warming, we're seeing much more change in the Central Coast, which from an environmental change point of view makes sense, because in the middle of a region, you tend to see a lot more turnover. You have things moving northward into it and things moving southward into it as well, possibly. So the central area generally tends to be an area of much more turnover in composition. The second step is that we'd like to aggregate data across space, time, and taxa. So aggregating data is basically aiming to make joint inferences from many data points whilst reducing the influence of any single data point in that set. So in a way, we're trying to dilute the effect of bias we're trying to dilute the effect of bias by aggregating together a set of records. And I'll show you an example here what that means, what this means here. Aggregating data can be thought of as reverse engineering survey structure. Remember I said that iNaturalist data are unstructured and that no one is mandated to take iNaturalist observations in a certain way. And so we do not have this standardized survey structure here that involves randomized sampling. What we do have is this, a set of observations over space. And what does it mean to reverse engineer survey structure? What we want to do is aggregate data in space and time, and as we'll see in taxonomy as well, to try and increase the information content of any specific data point. So what you're doing is you're trading off how specific a data point is. It's no longer a single GPS coordinate and a single timestamp. It's now perhaps a much larger area and a much larger period in time, but it has a lot more information in it. It has a lot more data points, and it allows you to understand a lot more what's going on in those areas at those times. So we're essentially generating some structure in the data by just looking at aggregates of these data. A specific example, a really important step in this process is to add the taxonomic dimension. So even if I'm interested in a single species and I want to know, for example, what the population changes in that species may have been, it's actually hugely beneficial to look at many other species as well that are in some way associated with the species I'm interested in. So if I'm interested in the blue triangle species here, actually that's not the only species I want to look at on iNaturalist to try and tease out ecological signal in that species data. I also want to look at the red square and the orange circle species because those species are going to do two things. They're going to inform on ecological relationships potentially on the species I'm interested in. So they may be species that the blue triangle species feeds on or needs for habitat. And so knowing where those are or what times is probably going to tell me about the distribution and abundance of the species that I care about. The other thing that they do is not ecological, but it informs on the actual observation biases and process. So if I look at many species worth of data, I'm going to get a much closer picture of what the overall observation effort was. I'm going to get that overall increasing curve over time, for example, and not just what that single species I'm interested in looks like. So let me give you detailed examples of this. Let's say my blue triangle species is Pysastor ocraceus here, the ochre sea star, emblematic species on the intertidal coast in California. And that's its pattern. It's going up over time, as we would expect, from 2012 to the present, or to last year anyway. If I look at these species here, so that's the species, remember that's the red square, the orange circle. Those are species that tend to be observed together with Pysastor. So that's in the same places at the same times. And perhaps they're related species. Perhaps they are other echinoderms, or they're species that Pysastor feeds on. So maybe they're also ecologically related and not just happen to be observed at the same time. But a lot of those are species that are common on the intertidal and that people tend to take a picture of if they take a picture of Pysastor. So what we want to do here is look at these two curves with respect to each other to actually get some signal of how Pysastor has been changing over time. So when we do that, we look at the number of times Pysastor ocraceus was observed divided by the number of times associated species were observed. We get this pattern here, which is much closer to the ecological signal we would expect for Pysastor ocraceus. That is, it was doing fine in 2012 up until between 2013 and 2014, the population decreased drastically as a response to sea star wasting disease. That lasted a couple of years, and then we're seeing some recovery in recent years. This is throughout the California coast. So this looks about right in terms of ecological signal, and we're using just iNaturalist data here. We're just looking at a lot of it. So how is this useful for management? We're trying to connect these data back to management. And what we're finding is that this is really useful as an early warning indicator of biodiversity change. So this is another example here. Pysastor is a great example in itself. But for example, the red abalone, which is a commercially important species that people harvest, has seen huge declines in the subtidal. So that's a little deeper in the water column. Generally, that's not really being observed on iNaturalist. What is being observed are the tide pools and the nearer shore. And what people are seeing is actually an increase in red abalone on the shore. And generally, unfortunately, it's red abalone that aren't doing too well. And that is because red abalone is more recently seeking refuge on the shore, so in shallower waters. And that means that people are seeing it more frequently. So it's an indication of potentially migrating to refugia, and actually the population maybe not doing so well. This is another example here, and this is a screenshot for some of the tools that we've been working on. And this is Hopkins Rose, beautiful nudibranch that many of you love, no doubt. And what happened to Hopkins Rose is that people started seeing it more and more frequently between 2014 and 2016 here. Specifically, 2015 was a huge increase. And so this is the black line here. And what you see here in gray is actually the variation in maximum temperature, rescaled here so that it fits on the same graph. But you can see that the pattern of it is pretty similar. So Hopkins Rose actually followed closely an increase in temperature, which we talked about, the increase in temperature along the California coast. And one of the beneficiaries of that was Hopkins Rose. So another example here of how this is useful for management is to try and understand and monitor change in ocean conditions and changes in populations that reflect those. So just how good are those data? Obviously, if we're just looking at iNaturalist data, it's a little bit hard to know exactly whether those trends are exactly reliable or not. I mean, those look about right in terms of what we know about the ecology of species and areas. But what you really want to do is ground truth these observations with maybe some data from standardized surveys, which, as we were talking about, is the accepted way that professional ecologists go about sampling areas and time periods. And fortunately, and pretty remarkably, some patterns are amazingly close to standardized survey data. So here these are just a couple of examples, but in blue here, you see the trends that I've been talking about in the proportional rate of observation on iNaturalist, or the reporting rate here. So that's that curve I was showing before. And in red here is data from standardized monitoring from marine. It's an intertidal long-term monitoring that actually helps the state monitor its marine protected areas in a standardized way. As you can see, if we look at, for example, Paysaster again, specifically on the south coast, I mean, that's almost 100% agreement between the two curves. Obviously, the units are different, but we're looking for the general pattern here. And as you can see, there are other types. This is another sea star here. There are other types of fluctuations that seem to be generally pretty well matched. There are always going to be differences because there are differences in scale between these two sets of data, but generally they're pretty good matches. Unfortunately, sometimes the matches are not so good. And so these are just a few examples again. But just to say that sometimes it doesn't work so well. And actually, we haven't been able to look at a huge number of species here. So we've looked at probably 15 species or so, because there aren't that many that are monitored in a standardized way. But we've tried to look at why sometimes trends match and sometimes they don't. And what we're starting to see is that the reason is probably a mix between iNaturalist observation effort and actually something going on with the ecology of a species. So, for example, there is a relationship, even though it's not so strong, between how well the trends match between iNaturalist and standardized surveys and the mean population size as assessed in standardized surveys of a given species. That means that generally, if the population size is higher, the trends will match a little more. And that's because probably people find it easier to find that species and take pictures of it, and it's generally recorded more comprehensively. It's not always the case. So rare species actually can match pretty well or very badly. So there's probably a range of rare species here that are rather species that people look for specifically, and these may be some of the more expert iNaturalist users, or just very rare species that are hard to find unless you're really looking for them in a very standardized, specific way. And the other thing is, unsurprisingly, there's probably a relationship between how many observations of a species there are and how good the trend is from iNaturalist data. I'm going to press on here. The last two steps that are useful in modeling iNaturalist data are really the more complex ones. And the first one is that actually, in those trends that we were looking at, there are probably still signals of bias mixed in with the ecological signal. And especially if we want to look at things explicitly in space, we're going to have to try and model the bias process itself. So that's going back to that equation. Don't worry too much about what this is, but these are bias predictors. So we want to include variables that can tell us a little bit about this bias function here. Why is it that actually we get this biased gradient here? And so those are things that we were talking about before. axis human population density. So I just want to give you an example of actually how rich iNaturalist data are, even though some things aren't, the survey effort isn't explicitly recorded and things aren't done in a structured way. But from the basic metadata that you get on iNaturalist, you can actually get a whole lot of complexity. And so here is 2 examples of 2 visits. So once again, that's a single user in a single spatial radius, about a couple of kilometers squared, in a single day. And these are the observations that they took. So we have how far between observations they walked on the y-axis and how long it took them to take those observations on the x-axis. And this is what we would call a low intensity sampling survey or visit. And that means this person took 3 observations and walked about a little more than 100 meters, and it took them about 20-25 minutes to do. However, on the right here, we have a high intensity sampling event where the person actually took upward of probably 100 observations here, so certainly several dozens of observations, and walked about 1,000 meters or so, so that's a kilometer or so, and took about 200 minutes to do that. And you'll see that a lot of them were within a short space interval and a short time interval as well, but then, there were a bunch of others that took some walking and some time. So these are some of the things that we can start including to try and understand what happened in a given visit. Why did people only record a couple of species? Was it because there were only a couple of species, or was it actually because they didn't look that comprehensively or exhaustively? Those things are going to help us understand the ecology more than the actual observation behavior. And so what we want to move towards is a type of model that actually models the ecological process, what we're really interested in. So that's how variation in temperature or habitat or variation over time affects the abundance of a species, separately from the observation process. So that's how the behavior of observers was affecting what was observed. And so that might include how many species were recorded, how far the person traveled, and how long they spent searching. Ideally, these things would be included in a model, and maybe they would be recorded at the onset. But you can derive some of this complexity from the data as they are. So I'm not going to show you any examples of just this type of model, but I'll point you to these 2 publications where we actually use these kinds of models, not just for citizen science records, but actually for natural history specimen collections as well. As I was saying, those data are very close in terms of the type of sampling that generates them. So what I do want to talk about is the last step, which is really probably the most desirable way to account for bias, is actually to recognize that iNaturalist data are incredible and they're incredibly rich, but they are no substitute, or rather there's no reason why they should be used by themselves when there are also standardized survey data available. There is no reason why those 2 data sources should be used individually and independently from each other. Actually, the real strength is to put them together and combine them. So these modeling frameworks have arisen more recently that actually allow you to take advantage of both types of data in a single model and allow you to make joint inferences. So here we have our presence-absence data from standardized surveys, which we can think of at face value, and then the citizen science data that includes presence-only data here. And we want to join our simple linear regression here that only includes ecological predictors together with our model for presence-only data that includes both ecological predictors and bias predictors, all together in one model. When we do that, and we have done it for a number of species on the coast for which we have both sets of data, we can get fairly detailed estimates of species distributions and variations in their density across space and across time. And we can also get detailed relationships. Those are modeled relationships, so sometimes they might look a little wobbly, but they give you the general estimate of how the density of a species changes with some environmental gradient, with some variation in the environment. So in this plot here, you have how the population density of Tetraclida rubescens here, the pink volcano barnacle, changes as a function of minimum temperature. And as you can see, the density of the barnacle increases as the minimum temperature does, right? We only have, we don't have the full range of the species here because we're limited to California and the US West Coast. But you can see that normally as it gets warmer, the density should get higher. And so it's more of a southern species. What that means is that we've been able to also model over time. Once again, these are inferences that are based on both the integration of iNaturalist data and standardized survey data. We can actually model a northward range expansion in the species. As temperatures gotten warmer, you'll be able to find these barnacles in higher densities up the coast, and certainly from the South Bay to the North Bay. So just to conclude here, all of these data analyses and patterns have been done by consulting. So the aim here is to be useful for management. So we've, from the get-go, consulted with stakeholders on the California coast and asked them what they would want to get from these data. What are the patterns here that are going to be more useful for them to try and understand the things that are needed? And so the stakeholders are not just managers at the local, state, and federal level, but they're also tribe representatives, researchers like us, the fishing community, divers, and members of the public who maybe are iNaturalist users and they're just interested in inferences from iNaturalist data. So I think I have a couple of minutes here. I don't want to take too long, but I just wanted to show you, maybe I'll show you the second one here. Ideally, I would give you a live demo here, but we've been putting together all these data in interactive form online for managers to go and see. So this is, for example, those patterns in the reporting rate of the Orca sea star that we were looking at. You can find those online, and those are updated as new iNaturalist data come in. You can also see a map of density based on those inferences that we were talking about and how the density changes in response to features of the environment like temperature and salinity. And finally, you can have a look at how density, model density, has been changing over the last 7 or 8 years or so that we have good enough iNaturalist data by comparing individual years here. So I will be forwarding a link to these tools really soon. We're sort of releasing them next month, May 14th. We're releasing them for the stakeholders. So I will share them with everyone. For the time being, if you're interested in finding out more and accessing these tools, please send me an email. And this is the end of the presentation. So I'll put up my email. Can I stop the presentation here? Anyway, please send me an email, and I will try and respond to you with any questions you may have and give you a link to those. But hopefully that gave you a good overview of what I've been up to for the last little bit and where we're going with this. Thank you so much for tuning in. And if we have a little bit of time, I'm happy to take any questions. hopefully you heard all of that. I have no idea.
Yeah, I think it was all right. I mean, I had some technical difficulties too, but I think it was on my side. So it was great. I think there's one question for sure. So if you guys have questions, put a question mark in the chat box or type your question in, and I'll read the one that we have. It was from Kenichi. He was asking about validating naturalist-based population trends and wondering if you tried other taxa, perhaps taxa that are not involved in kind of the semi-structured kind of efforts like we do at Pillar Point. Like, could you just subtract all the Pillar Point data? And if he's still on, he can maybe clarify a little bit, but that was his question.
Yeah, no, I mean, sorry, Kenichi, you can weigh in if you want, but I think I got the gist. Yeah, okay. So, yeah, no, absolutely. So that makes a lot of sense. I think, obviously, Rebecca and Alison have done a terrific job in Pillar Point, which really approximates, it doesn't approximate, it is basically a standardized survey, but some of the metadata aren't recorded in a standardized way, but the information is all there. However, we do it regionally as well. So I've shown patterns regionally, and I'm not aware of similar efforts happening in Southern California, but for example, the best correlation I was showing between trends was actually in Southern California for Pysastes. So there is certainly evidence that even in areas that do not involve Pillar Point, there are very good correlations. But once again, it does vary a lot. So there's so much going on there.
Awesome. And it looks like Shannon has a question too.
Hi, Gio. That was really fun. So as a microbiologist, I can deeply appreciate that you need specific structured surveys and how they can be partnered with iNaturalist surveys. And I think it's a perfect complement, especially as I want to track disease organisms over landscapes that are very dynamic in host composition. And I was wondering, in your interactions with the different stakeholders and communities, if there is a talk about integrating genetic or genomic data into this individual population level framework.
So should there be talk about it? Yes. Like, sure. Like, I totally believe in it. I think if we're being honest here, I think from a management standpoint, we're so far from it, at least on the coast. But that's not to say that that's the case everywhere. I'm talking not about collecting genomic data, but about thinking of it in terms of integrated with these other kinds of data, right? So that's what we're very far from.
Yeah.
So unfortunately, we've certainly had no conversations about it, and it's not really even on the cards at this point. But I think, eventually the dream is meta-barcoding with citizen science, with standardized surveys, with museums, I mean, that's where we should be going in terms of big data.
Thank you.
And thanks to Shannon. I think Natalie has a question next.
Yeah, so you showed the New Phytologist article looking at herbarium specimens and the bias in them. Has there been any studies comparing herbarium data with citizen science data, and maybe ground truthing things or anything?
So I'm not sure about it, to be honest. So not that I know off the top of my head, but also because most people just get their data from GBIF and just take whatever species occurrences they need and make sense, regardless of the data source. So very little has there been work done looking at the different inferences that you get at different types of data that come from the same big data warehouse. So I'm not really familiar in it. I know there's a lot of gap analyses in GBIF data and how reliable they are, but generally they combine both types of data. So not that I'm familiar with, I can have a look, but that's certainly something that would be useful to do.
I think, so just to follow up, I think it would be really interesting because botanists really hone in on those really rare species. And so it's interesting to see whether citizen science also captures those rare species.
Right.
Maybe that's an SSI.
Absolutely. And as you've seen, I think in terms of rare species, there's real variation in how well they are recorded on iNaturalist and how reliably they're recorded. And I would say probably the strongest strength of iNaturalist is actually probably the less rare species, but not so common. So somewhere in between. Yeah, so they're hugely complementary for sure.
Thank you.
All right, the next question is from Darrell, I think.
Hi, everyone. That was a great talk, Gio.
Hi, Darrell.
I'm just curious, thinking really, really broadly, is there one or two things that any citizen science project or citizen scientist should be thinking about when going out? Is there some simple, like, rule of thumb or anything general that you can tell us?
That's a great question, and I don't want to be prescriptive and too prescriptive. So the first thing is go out.
Gio, did you hear Darrell's question?
I did. Yeah. Sorry, what did I cut out for a second? Yeah. Yeah, I was saying that I don't want to be too prescriptive. I don't like to be too prescriptive, but the first thing is go out, like get the data because they are useful, no doubt. And then what I would say is, can you hear me still? Oh yeah. What I would say is, clearly the more comprehensive a survey, the better. So maybe move towards a higher intensity sampling event out of the events that we were looking at, rather than a low sampling event. So maybe record a few more species, maybe take a little longer, walk a little further. Those events are likely to be more useful than just taking 1 or 2 pictures. But if you can only take 1 or 2 pictures, take only 1 or 2 pictures, because those will be amazing compared to no data whatsoever.
Thanks a lot.
All right, I think it's just after 4 o'clock, so I think we'll take a break there. I don't see any more questions. So if you have more questions, feel free to email Gio and let him know that you have some questions and he'll follow up. And if you want those tools before they're released, send him a note and so you can play around with them. And Gio, thanks so much. It was really, really great. If people can stick around, or I guess, is it sticking around on the same link for the happy hour? I didn't do it last week, but I think that's the case. All right. Thanks so much, Gio.
Thank you. That's awesome. That was fun. All right.
Thank you so much. It was great.
Thanks, Gio. Awesome.
Thank you all. Take care.
And there's a separate Zoom link for the IBSS happy hour.
There's a separate link. So go to your separate link for the get yourself a drink for the happy. And
See you at happy
remember
hour.
to join later for the gala. If you haven't.
All right. Awesome. And go out, make some observations for the
All right,
city.
Gio, that was great.